algae section, we have studied already the general characteristics, the economic importance, the classification of algae, and also the reproduction methods of algae. And in algae, we have also studied the life history of nostoc. And now we are studying about the next in Chlorophyta Chlamydomonas. Basically, we have studied the structure of the Chlamydomonas cell. So, Chlamydomonas is a green alga, which is found in fresh water and also in the terrestrial regions. Now, Chlamydomonas is a unicellular alga. So, this Chlamydomonas, which is unicellular, it is made up of a single cell, and the chloroplast, which is a somewhat cup shaped like this. So, this is the shape of the chloroplast, which is cup shaped, and the nucleus is present in the center, in the depression part of the. So, this is the chloroplast. So, these are actually the blepharoplasts. They are small granules which gives rise to the flagella like this. And these two blepharoplasts are connected by a structure called as plasmodesmos. Now, this blepharoplast granules, they give rise to the flagella, which are usually directing backwards. Then the cell is surrounded by a cell wall, and this cell wall is mostly hemicellulose. So, this hemicellulose cell wall, which encloses the cell membrane and the cytoplasm. The cytoplasm contains a cup shaped chloroplast, and in the depression part of the chloroplast we find the nucleus, and in the chloroplast we are having the stroma fluid, and this stroma contains the thylakoids. So, the thylakoids are flat membranous structures which are almost parallel to the cell wall. They are arranged parallelly to the cell wall, and the thylakoids contain the chlorophyll pigment. In addition to that, the chloroplast also contains pyrinoids. So, pyrinoids are small spherical bodies which are actually proteinaceous. So, these proteinaceous bodies are surrounded by a layer of starch, a sheath of starch, and they are present in the chloroplast. So, there may be one or two or more pyrinoids in a chloroplast. And as usual, in the anterior part, here there is a structure which is called as the eye spot. So, there is a red colored or somewhat orange colored structure, which is actually a pigment. It is a orange pigment structure or somewhat reddish pigment which is present at the anterior part of the cell. So, this is called as the eye spot. So, the eye spot is a photoreceptive organ. So, that means it is very sensitive to light, and it helps in the movement of this cell. As the flagella are present in the cell, so the cell is motile. It is having the power of locomotion with the help of this flagella. So, the direction of the cell is actually done by this eye spot. So, that directs the cell in its movement or in its motion. Then the cytoplasm, which is surrounding the chloroplast, contains all the cell organelles like mitochondria, Golgi complex, endoplasmic reticulum, ribosomes, and all that. Even ribosomes are also present in the chloroplast and also freely present in the cytoplasm. That is about the structure of the this Chlamydomonas cell. So, the Chlamydomonas cell, it is a unicellular structure which is usually present and it is found in the fresh water as well as in the terrestrial regions. Then if we come to the other structures of the cell, so this is the paradesmos which are connecting the one, and the eye spot is also connected to the centrosome in the cell. This is the cell structure of Chlamydomonas. Now if you come to the reproduction of Chlamydomonas, how the reproduction takes place, so which is the main part of this life history of this organism. So reproduction takes place by asexual and sexual type of reproduction. So in asexual we come across the zoospore formation. So the formation of the zoospores is asexual reproduction, and also a planospores, that is also another type of reproduction, a planospores. Then we are having the Pamela stage in this asexual. So Pamela stage is another method of sexual reproduction. Now one by one, in a zoospore formation, what actually happens? So in a zoospore formation, suppose this is the Chlamydomonas cell. Now it is by simple cell division, zoospores. The cell, the Chlamydomonas cell divides longitudinally like this to form two cells. So first one cell is there, so then the cell will divide into two, and the flagella may be lost or may remain as they are, and however the blepharoplasts also will divide, and again the flagella will increase. So here the two cells are formed in case of zoospore formation. Now when by just a simple mitosis division, after two cells are formed, again these two cells will divide perpendicular to each other. Now suppose this is one cell, if it divides into two cells in this plane, two cells are formed. The next division will be perpendicular to the first, also longitudinal. So four cells are formed after division. Now these four protoplasts, each protoplast will develop, they will separate, and they become four cells like this, and they develop the flagella and also nucleus. So the nucleus is developed and also the flagella is developed, and they become four independent cells. This is called as zoospore formation. So zoospore involves the cell division process. So four cells are formed, all the four protoplasts, they differentiate and they develop into individual cells. Each cell will develop its own flagella and all that. Even the chloroplast also will divide. After the division of the chloroplast also will develop. It will grow in size according to the cell, and it will become an independent cell. That is in the case of this zoospores. So actually the zoospores are separated, they are liberated freely, they are released freely into the region, into the water or like that, and then like this one cell develops into four cells. Then a planospores. So when there is scarcity of water, here what happens, sometimes the cell will come to rest. So the cell will not divide, it will lose its flagella. So the flagella are lost, and the nucleus and all they remain as it is, and they store some amount of reserve food material, and these this protoplast which is present inside, this reduces from its cell wall. This is the protoplast which somewhat contracts or withdraws from the cell wall and it remains inactive. So this is called as a planospore. So a planospore means here there is no division. The existing cell, instead of losing its existence, it will remain till favorable conditions arrive. So till the favorable conditions, these structures called as planospores, which are present or which are formed in the cell, they develop into the Chlamydomonas cell during the arrival of the favorable seasons. And sometimes there are also called what are called as this Hypnospore. Similar to this, we come to this Hypnospores, but these are little different from the Planospores in the sense Planospores they do not develop any cell wall. So there is the existing cell wall, but inside the protoplast will withdraw from its cell wall and it is somewhat contracted, condensed in other words, and remain inactive till the arrival of the favorable season. But in the case of Hypnospore, the cell will develop a thick wall which becomes resistant to the environmental conditions. So this Hypnospore develops a thick cell wall and thus it will develop into an independent Chlamydomonas cell after the arrival of this favorable conditions. Now the next is the Pamela stage. In the case of Pamela stage, here actually this is also a kind of asexual reproduction. Here actually the cell, suppose this is the Chlamydomonas cell. So this cell will divide by mitosis to form 4 or sometimes 8 cells like this, same as that of zoospores. But here the zoospores are not formed there. So the protoplast will divide like this, and these cells will separate actually. So the 4 cells will separate and they remain like this. Sometimes again these cells may divide to form, suppose this single cell will divide to form another 4 cells group like this. So this also divides to form another group of 4 cells. So these cells are formed here like this. But one important thing what takes place here, after the formation of these many cells, these cells are enclosed by a mucilaginous sheath. A mucilage sheath is formed in this cells, surrounding the cells. So the group of cells which are formed, so they are found in groups like this, in the groups of 4, or sometimes we may find groups of 8 cells. Generally 4 cells are formed in this, enclosing this mucilaginous matrix. So this structure which is seen in the groups, they are not having flagella, but they are having cells. They are found only in groups. So they resemble an algae which is called as Pamela. So Pamela is a name of a alga, green alga. So the reproduction stage or this stage of reproduction resembles a alga called as Pamela, hence it is called as Pamela stage. So but these cells which are non-motile, they are not having any flagella. So these cells become motile during the arrival of the favorable conditions. So these cells remain as they are, just like how the Hypnospore or the Planospore remains without any activity. So these cells which divide, they become well protected in the mucilaginous matrix, and then the cells they form in the group of 4, and all these cells they appear like the structure of a Pamela algae, hence it is called as the Pamela stage. And at the arrival of the favorable conditions, these cells will develop a flagella and then they are released from the mucilaginous sheath. That means, so how many cells are there in this Pamela stage? All the cells will develop into an independent Chlamydomonas cell. So the Chlamydomonas cell will develop independently and then they are released from the mucilaginous matrix. So that is about this Pamela stage. So these two stages, they are usually whenever there is a kind of desiccation, scarcity of water. But Pamela stage, it is at the time of partial desiccation, means not complete absence of water or like that. So when there is partial desiccation, Pamela stage can be seen in the case of Chlamydomonas. So that is about the asexual reproduction. Now if you come to the sexual reproduction, the second one, here all the three types of sexual reproduction takes place. So there is isogamous type of reproduction, there is anisogamous type of reproduction, and also there is oogamous type of reproduction. So these three type of reproduction takes place in the Chlamydomonas animal. Now in the Chlamydomonas green algae. Now how this isogamous, anisogamous and oogamous takes place. As we already studied in the algae, isogamous means the gametes are similar and the gametes are not similar. Here there is well defined structure where gametes are produced. Now the gametes which are produced, the male gametes and female gametes, sometimes they are surrounded by a cell wall or may not be. When there is no cell wall in the gamete or when there is cell wall present, so these are called as gymnogametes, and when there is cell wall, these are called as calyptogametes. So here usually calyptogametes are seen in the Chlamydomonas. So generally when gametes are not surrounded by a cell wall, gymnogametes, when the gametes are surrounded by a cell wall, they are called as calyptogametes. Here the gametes are same, the gametes are similar, isogamous. Here the gametes are not similar. That means in isogamous what happens? When we observe the gametes, we cannot identify whether it is a male gamete or a female gamete. These gametes, suppose this is a male gamete and this is a female gamete. So both gametes appear morphologically similar. There is no any differentiation, or we cannot differentiate between the male and the female gamete. So this gametes which are produced in isogamous, here again we come across two types of species, homothallic and heterothallic. So homothallic and heterothallic type of species are found in this isogamous. In homothallic what happens? Now suppose this is a single cell. Now this cell divides to form 2, 4, 8 or 16 cells inside. So these cells which form cells like this, now they form gametes. So some of them are male gametes and some of them are female. So the gametes are produced from the same cell. When they are produced from the same cell, it is called as homothallic. But however, both gametes are same. So the male gametes and the female gametes are same. But however, here the word male and female is not used. Instead of using that, plus type and minus type of gametes are it is called as, because male and female word is used whenever the male reproductive system or the female is well developed. But here there is no any clear distinction between the male and female. But this male and female, which we call it as plus or minus, we call it when these two fuse each other. When they do not fuse each other, it means that both are same. Either they are minus type or they are plus type. So when the two gametes are brought together or they are in contact with each other, when they fuse, one is of the plus type, another of the minus type. And which is plus or minus, that is only known by chemical nature, but not by the morphological nature. So here the male and the female, which generally we can refer, so this are produced by the same cell. A single Chlamydomonas cell give rise to the male and female gametes, and these male and female gametes are of same type, and each Chlamydomonas cell gives rise to about 16 or 32 or even 64 cells are produced here. So 2, 4, 8, 16, 32, 64, up to 64 cells are produced. All these cells, they are motile, means they become the motile cells, and these are usually gymnogametes. In case of this homothallic species, we come across gymnogametes, and then this two gametes which are formed, suppose this is a plus and minus type, so both of them will fuse. So when they fuse each other, they form a single cell which is having 4 flagella like this. Quadriflagellate it is called as. So these are biflagellate. So when these two fuse, all the 2 pair of flagella, they remain in a single cell, and this is called as the zygote. So the zygote further develop into a structure called as Zygospore. So this Zygospore actually does not have any flagella. So that means it forms a thick wall like this. It develops a thick wall around it, and this is called as the Zygospore. The Zygospore will further develop. So how that Zygospore develops, we shall see after studying these 3 types. So this is homothallic. A single cell produces gametes. So up to 64 totally. Out of 64, 32 are plus type and 32 are minus type. 32 male and 32 female. So they fuse to form 32 zygotes, 32 zygospores. Suppose there are 32, totally 64, 32, 32. So these fuse and form 32 zygotes. 32 zygospores are produced from this single cell. But in heterothallic, 2 separate cells produce. Suppose this is one cell. Suppose this is another cell. This is plus type and this is minus type. So these also will produce gametes. These also will produce gametes. So when they produce the plus type and minus type, again it forms the zygospores. They are isogamous, but the gametes are produced from separate cells. Plus type from one type of cell, minus type another type, means the male type and the female type cells are from separate. They are called as heterothallic. So that is isogamous. Gametes are same. Here also the gametes are same. These are the male gametes, and these are the female gametes like this. So these will fuse and form the zygote, and the zygospore is formed. That is isogamous. But in anisogamous, what happens? The cell will divide, and the gametes which are formed, they are of different size. Again here the anisogamous, usually the cell, suppose this is a cell and this is another cell. This is plus type and this is minus type. Usually in anisogamous, heterothallism occurs. So the male gametes are somewhat smaller. The female are somewhat larger gametes. So this male which are formed, that is the plus type, they are called as the microgametes, and these are called as the macrogametes. So microgametes are small and macrogametes are larger, and these gametes are usually calyptogametes. This we should remember. Already I mentioned, if there is no cell wall, gymnogametes. So in isogamous they are usually gymnogametes. When the cell produces the same type of gametes, they are having no cell wall. But when the cell is dividing, or when it is isogamous type of reproduction, that means the gametes are of different size. We can differentiate morphologically which is male and which is female, or which is plus type and minus type. So the smaller gametes are male, they are the microgametes. The larger are minus type or the female, they are called as the macrogametes. So this macro and micro, again they will fuse and form the zygote like this. Same. So the two cells, the small and the large, they fuse and form the zygote. So the zygote then develops a thick cell wall and forms the zygospore. That is in anisogamous type of this reproduction. In oogamous what happens? It is somewhat different. So in oogamous, here also it is heterothallic. Suppose this is the male cell and this is the female, which we refer it as plus and minus. Now this divides to produce many gametes like this. It divides to form gametes, but here we don't refer it as micro or macro gametes, but in case of this we find micro and macro gametes. Of course we can differentiate here. Usually these are smaller in the size, but this female cell forms only a single gamete. It does not divide. In case of oogamous, the female cell will lose its flagella. It will lose its flagella like this, and after it loses its flagella, it forms a single gamete called as the macrogamete. That means it is usually larger than these male gametes. That is why here we can refer it as macro and micro. Here both are having flagella, the smaller male and the larger female. Here also the male are smaller, the female is larger, but it is without flagella. It loses its flagella and just the protoplast just becomes spherical shape, and it forms the macrogamete and becomes non-motile. That is the thing. So the this macrogamete becomes non-motile, and it is called as the egg or the ovum, and then this male will produce about 16 or sometimes 32 microgametes, and these microgametes, they are released into the water, and usually these microgametes, they will move towards the macrogamete. The macrogamete does not move. It is usually non-motile, and it is non-motile. So these will move towards the macrogamete, and then they will form the zygote by a fusion, and then the zygote develops into the zygospore. So like this, whether it is isogamous, anisogamous or oogamous, so gametes are produced similar, gametes are different. Gametes are also different. Usually the female is macro, non-motile. The male is motile. So zygote is formed, zygote and the zygospore is formed. So when the zygospore is formed, now this zygospore will divide. Suppose this is the zygospore. So the zygospore will divide by meiosis. Actually the zygospore contains reserve food material. It contains large amount of fat material, some reserve food material, and it is somewhat orange red in color due to the presence of the pigments. Now after when the zygospore starts developing, so this orange or red colored zygospore, it turns to green in color. So this green colored zygospore, so it is the indication that the zygospore is initiating or start to develop further, and then the zygospore undergoes meiosis. So it undergoes meiosis to produce 4 nuclei. Now these 4 nuclei which are formed, suppose this is the cell, these are the 4 cells which are formed inside. So these 4 cells which are formed by meiosis, so those 4 cells which are formed by meiosis, they are called as the meiospores or gonozoospores. They are called as the meiospores, or sometimes they are also called as the gonozoospores. They are called as. These gonozoospores which are formed, they are 4 in number. So each zygospore produces only 4 meiospores or 4 gonozoospores. So these gonozoospores are released by the breakage of this zygospore wall. As I already said, the zygospore wall is thick, so it is having a thick wall. So after the zygote develops into zygospore, it develops a thick wall around it. So that zygospore wall will break, it will crack, and by the breakage of the wall of the zygospore, the meiospores are released, and each meiospore, actually it is not having a cell wall, this is only cell membrane. So these meiospores, when they are released, so they become independent and they develop a new cell wall. They prepare its own new cell wall, that is hemicellulose, and they develop a pair of flagella, and they develop into 4 Chlamydomonas cells. That is how a single zoospore develops into 4 Chlamydomonas cells. So whenever the potentiality of the cells they decrease, they conduct such type of reproduction, and so the zygospore divides by meiosis to give rise to 4 meiospores or gonozoospores, and these zoospores are simple cells which are surrounded by a cell membrane, and they are released and which are surrounded by a cell wall, and each develops a pair of flagella, and then that is how the 4 Chlamydomonas cells are produced in this reproduction of Chlamydomonas. So that is how the Chlamydomonas cell, which is unicellular, so it is having the stages of asexual reproduction and also sexual reproduction by isogamous, anisogamous and oogamous reproduction. So that is about the life cycle or the life history of Chlamydomonas cell.